Hey everyone, I think it's time that we talked about our poop. There's not a lot of controversy about whether we have microplastics in our poop, but maybe for that reason it's more important that we talk about it because there are actually really good data here that teach us something. So there are a lot of studies about this. I'm going to pick one for today to go over in detail, and we'll see what we can learn from it. Before we do that, I wanted to give a little bit of perspective. This is a representation of how the field of microplastic has taken off in scientific literature. So this is the number of scientific publications per year that mention microplastics, and it's grown from essentially zero to over 60,000 last year. In fact, you can fit a function to that, and this is probably the best data fit in the entire field of microplastics. There's an R squared of 0.996. So what really made this take off? Well, the term itself was first used for its current meaning in about 2004. Before that, it just meant, it was just people describing microstructures of plastics, and the word would pop up occasionally. But it started to mean particles of 1 to 5,000 microns in about 2004. But one could argue that it didn't really take off like crazy until about 2018. And that was when the first report came out on microplastics in human stool. And I think that sort of sparked a lot of questions and curiosity and imagination about what those microplastics might be doing between the time they come into our body and the time they exit the body. And so a lot of this growth has been a big race to be the first to report microplastics in various organs within our body. And sadly, accuracy has not been necessarily the greatest predictor of success and widespread attention within that effort. But nonetheless, there have been a lot of publications, and I think this first discovery of microplastics in human stool was a big driver of that. The public interest has grown as well, and kind of the same sort of trajectory. So this is from Google Trend, the number of web searches for microplastics. This is on a relative scale, so the axis on the left is not applicable to the red line, but it's just to show you the same sort of pattern of growth. And the public consciousness has sort of mirrored the number of scientific publications. And of course, it's always fun to do things like this, so the number of total scientific publications is growing essentially linearly, and the microplastics mentions are growing in a sort of a power law. And if you extrapolate those out, every scientific publication should be a microplastics publication within about 10 years. So it's incredibly rapid growth for microplastics related publications, and I'm sure it'll start to level off, but it's always fun to do extrapolations like this anyway. Okay, this is the publication we're going to go over in detail today. It's pretty recent. It was just published last year. The main thing that I want to highlight from this is that they're trying to find a link between the microplastic content of our stool samples and our behavior with regard to handling and use of plastics, okay? And so the take-home message they have here is that the different scenarios did not result in a consistent pattern of microplastics. However, the use of plastics for food packaging and preparation and the consumption of highly processed food were statistically significantly associated with microplastics content in stool. That's kind of a mouthful, and hopefully that will make sense to you by the end. So obviously, I don't just jump to the conclusions. I want to see how trustworthy and valid their results are. They did a lot of the same things that just about everybody does in this field. They used non-plastic materials and cloths, cleaned equipment and surfaces rigorously. They used a laminar flow bench to keep plastic particles from settling in their work areas. But they kind of did some extra effort. They went the extra mile on a number of things. So their lab was in a dedicated, isolated, clean, windowless room. Okay, they only used it for this kind of work. They have a separate ventilation system for this lab so they don't get dust and particles coming in from other parts of the building. Restricted entry for specially trained staff, so only people who know exactly what they're doing go in there. You don't have people bringing their little microplastic clouds into the area willy-nilly. They have clean room sticky mats to sort of pull some of the microplastics off their shoes that they might have brought in. And then they also perform their analyses blind, so the analysts don't know what they're measuring, so they can't sort of try to impose their own ideas about what they should find into the results. So this is really sort of top of the field in terms of controlled environment for performing the experiments. In addition to that, they have very strict guidelines and criteria about who was eligible to volunteer for this study, and they asked a lot of their volunteers in terms of record keeping. We'll go through a couple things. So these are some of the inclusion and exclusion criteria for their volunteers. And then they had this lengthy questionnaire, protocols, and even a description of how to take a blank. So within their own bathroom, could there be any contamination from the environment there that could come in? So they have field blanks that they ask their participants to generate. Okay, I'm not going to go through this in detail. There's a lot here, but the point is that they were very thorough, and they have a lot of information about the participants as well. Okay, so let's start looking at their results. But before we do, I always like to look at what they did for their controls. The procedural blanks just mean they did exactly the same thing that they do to their stool samples. They just left out any stool. And in some cases, they still see some microplastic particles. In fact, there are a total of 5 particles found in the 4 blanks that they performed. This 0 here, there's actually one. You can see in the footnote that they found a polystyrene particle. And they did a good job. So they corrected for these. So they assumed that anything in the blanks would also likely be in their experimental samples. And so they simply subtract that out. Now, the reason this is a 0 in this one case is because none of their experimental samples in that set that this C blank was used as a control for, none of those had any polystyrene in them. So you can't assume that there's a negative number of polystyrene particles in a stool sample. That doesn't make any sense physically, but I think it points out a problem logistically with the way they do their blanks and the way I think all the people do their blanks. And that is simply, they just don't do enough of them. Because what this says to me is the assumption that whatever we have in our blank, we need to subtract that from our experimental samples because we probably accidentally got that same amount of stuff into our experimental samples. Well, that's obviously wrong in this case. They did 60 total samples, so maybe 15 of them were controlled by this C blank, and none of those had polystyrene in it. So I think you're dealing with such low numbers of particles that if you don't do any averaging for your blanks, you run the risk of doing incorrect corrections, if you'll pardon the expression, for the content of the blanks. So you're almost certainly undercorrecting for A and B, where you found 0 particles, and you're almost certainly overcorrecting for D, where you found 4 particles. In fact, I would speculate that it would be more accurate to just average all 4 of these and take that as your correction across all 60. But the real root of the problem here is you're just not doing enough blanks. You really, this is where the most uncertainty is, because you have such a small number that if you just rely on one blank to control 15 samples, you could introduce a real systematic error into your results. Don't know. I mean, I'd have to look at the setup and the flow, the experimental flow and everything, but just doing one blank to control 15 samples is not a good idea. I would try to do something like 4 a day or something like that, and that way you can kind of look for periodic trends throughout the lab as well. Maybe do 2 in the morning and 2 in the afternoon, and see if there are any temporal trends that start to develop, and see if your 2 that you're doing at the same time are generally the same or they're generally different. So you'd learn a lot about not only would you have more accurate data in the end, but you'd learn more perhaps about how to keep your lab even cleaner than it is. So I'm not a huge fan of this low number of blanks. It's what everybody seems to do in this field, if they do and report any blanks at all. But they really need to do more for this to be accurate. Okay, end of soapbox there. So in addition, I was very happy to see that they asked their participants to collect a field blank. So this is particles settled out in their bathroom at the time that they were collecting their first stool sample. And you can see that in this case, there are 62 particles in 15 blanks. And that's not surprising. Okay, this is a relatively uncontrolled environment. And so the fact that you have more particles found per sample is expected. What is odd to me is that this statement here, they didn't correct for these, and at no point did they explain why. And I contacted them and asked them several times, and one time they told me they didn't have enough time to answer me, and then after that they just didn't answer. So I don't know why they didn't apply this correction. I mean, why would you collect field blanks and then just not correct for them? So that's a mystery to me. It turns out in this case, it doesn't matter that much because their samples themselves are just filthy with microplastics. Okay, so in a 10-gram sample, which is about what they used, approximately 10-gram sample, they have on average about 57 microplastic particles. Okay, and so the field blanks averaged about 4 and the procedural blanks averaged about 1. So honestly, their data are going to be pretty good. I mean, better than average in this field of study, regardless of whether they apply any correction or not. They did collect for the procedural blanks. They didn't correct for the field blanks. And I'm not really sure why, but their data are valid nonetheless. But this brings up a kind of a side point. So if you'll indulge me in a separate discussion, sort of a review, if you look at some of the other studies that we've gone over in previous videos, okay, so here's the procedural blanks for the stool on the left, and then for the liver and for the lungs, they also collected and corrected for procedural blanks. And you can see that they can afford to do that because the number of microplastics in their actual samples were significantly higher, and so you could subtract those out and still have a number that you can report as a reasonable estimate of your concentration of microplastics in your tissue of interest. In contrast, some of these other studies, which I have not been a big fan of, the results that they're reporting, they either don't report their blanks or they don't report the results of their blanks, and certainly they never subtract out the values, their average values of their blanks. And their total concentrations in, for example, this first paper on the placenta, the one with the breast milk, and then the 2 urine papers, which I've gone over in previous videos, they're the ones that really need to be rigorous and very careful about subtracting out a correction for their blanks because they're down close to the noise and sometimes even a little bit below the noise. They're really about the same amount that you would expect to find if there's no tissue in there at all. So that's why I really struggle to trust the results from these studies. And you'll remember the breast milk paper especially had an additional source of contaminants, which is microplastics adhering to the skin, the pores of the nipples of the mothers themselves. So, really this is just additional evidence that you really have to be careful about trusting the results from these papers. And it's just striking to me that the ones who really need to be careful about doing the blank corrections are not doing the blank corrections at all. Just quickly, I'll mention that two of these papers, they reported not just the microplastics, but also pigments. So some of those pigments may have been associated with microplastics, but probably not. Okay, back to the study we're focusing on today. They had fantastic volunteers who really provided them with a lot of data, and not only that, but were willing to alter their lifestyles in order to see if those changes resulted in any measurable changes in the samples. And so what they did was, they just did their normal diet, whatever they normally do for days 1 through 4, and then they collected the first sample, and those median results are shown here. I show you both the value they have, and then the adjusted, the lighter one is the one that's been adjusted for the field blanks, because no one told me why I shouldn't do that adjustment, so I'm going to do that adjustment. As I said before, it doesn't make that much difference. So after this first sample, then they went to a low plastic scenario. Okay, so these next 4 days, they were supposed to really avoid contact with plastic as much as humanly possible in today's environment. And then after those 4 days of trying to avoid plastic, they collected their second stool sample. Okay, then they did a reset for 3 days where they just kind of went back to their normal diet and collected another sample. And then for the last 4 days, they tried to go to a high plastic scenario. Well, what you can see here, oddly, is that the high plastic scenario actually has the lowest median microplastics per gram in the stool samples after that. And so that's where this first half of this statement that you saw in the abstract at the beginning, the different scenarios did not result in a consistent pattern of microplastics. So that's what they mean by this. Like, I'm sure this is not what they expected to see. I wouldn't have expected to see it. I think it's just kind of random noise. It doesn't really look like these changes in behavior had much, if any, change in the number of microplastics going through their gastrointestinal system. Okay, let's talk a little bit about the second half of this statement. So the way they got to that was, it's a little bit of word salad here, but they did go through all that, the questionnaire and all the survey information and the food intake information, and they found some correlations that seem to indicate there's some correlation between microplastics and behavior. For example, during the normal diet phase, polyethylene seemed to correlate with the use of plastics overall from person to person. Okay, so that's just in that first stage, results of the survey correlating to microplastics in stool samples. And then they also say polypropylene seems to correlate with eating highly processed foods, but only when you look at the consumption data for the last day before the stool sample was acquired. And then the last one seems like the strongest one to me, the sum of microplastics detected, so the total microplastics correlated with the use of plastics throughout all of the normal diet days. So those p-values are low, so those are statistically significant correlations. But this feels a little like a fishing trip, like they were looking for correlations between, all the different types of plastic as well as all the plastic put together versus all the different behaviors. And so if you look for 100 correlations and you find 3 that actually have low p-values, it's hard to say what that means for sure. Now, I will say, I want to give the authors credit. They didn't oversell this. They did find these correlations. They may or may not be important as they come to larger studies. And they did call this a pilot study. So I assume there will be a follow-up with more participants, and maybe that will flesh this out. So can we control our own destiny in terms of microplastics consumption? I would personally put that somewhere to the right of probably around the maybe, but maybe closer to no than to yes. So I am curious to see what they find if you go to larger data sets and more volunteers, if these correlations hold up. Intuitively, it seems like this should work, right? It seems like if you expose yourself to less plastic, you should have less plastic going through your system. But maybe we need to rethink the entire thing. So that's why this is an important study to help us answer questions like that. Okay, we have valid data from this report. So what else can we learn from this? It struck me that they point out in this paper, and it's similar comments have been made in nearly every paper in this field, that only the small particles are important, right? The big ones just go right through us. They don't do anything. They just go in and they come out, okay? But if it's less than 150 microns, then you have a chance that it will pass through the intestine epithelium, which I had to look up, but it basically means it can get out into other organs. And then the really important ones are these micro and nanoplastics that are less than 10 microns because they can get to organs and they can even get into cells. They can get across cell membranes and into cells. So, but what we have is information in the 50 to 5,000 micrometer range. Okay, so it's of limited value for determining risk in these criteria, under these criteria above. Well, if you saw the last video talking about how much microplastic we ingest per year or per day, I forget, there's this handy equation in the review paper that we went over in that video, and I put a link to the paper and to that video here, where you can answer questions like this. Okay, so you have about 3 microplastics in this size range, but what we really want to know is how many microplastics does that mean in the 1 to 150 micron size range? And by simply plugging the appropriate values into this equation, and assuming alpha equals 1.6, and again, that comes from this review that I give you the link for, you can estimate that it's about 30, okay? About 31 particles are going to be in this size range, okay? So that's 31 microplastic particles per gram of stool that could be in the size range that could get out into other parts of the body. And then the question about micro and nanoplastics less than 10 microns, you can also answer that question. In this case, we're going all the way down into the nanometer scale range, but that's still answerable by the equation that you see on the slide. And then you're talking about a couple thousand. So a couple thousand tiny particles per gram of stool that are coming in. Whether that's enough to be a significant issue, I guess, is an active area of debate. But at least now you have that kind of information, which can be useful for people who know more about our biological systems. So another important thing that we can extract from these data, for me, is the mass balance. Okay, so in that previous video, we talked about intake of microplastics, and here we're talking about output. Well, those two should be very similar. So again, from this paper, they say, only about 0.3% gets into the body at all. The rest is just going in and going out. So the number and the mass of microplastics that we take in should more or less match the number and the mass of microplastics that come out. Okay, so again, we start with this median in the 50 to 5,000 micron size range and I'm using all the scenarios here from this paper. Now we're going to multiply that by about 130 grams of poop per day, which is just a number I got off the internet. And that gets us up to 381. Then we're going to use this fancy equation again to convert from 50 to 5,000 into 1 to 5,000, which is the conventional definition for the size range for microplastics. And that way we can compare that to the data from that review for the intake numbers. Okay, so now we're at 4,228 microparticles per day. And that is easily converted into about 1.5 million particles per year. And then we can estimate from that that's about 1 milligram per year. And that's assuming that the average weight to number ratio for this set of particles is the same as it is for the intake. Okay, so assuming the particle size distribution, the composition distribution, and the density distribution are about the same as they were in that intake study, that should give us about this number. Now, there are a lot of estimates in here, but again, it gives us maybe an order of magnitude sort of idea about output of microplastics from our body, and it's about 1 milligram per year that we're pooping out. Okay, so if you compare that to the intake from that previous video, it really matches remarkably well. I'm almost embarrassed to show this to you because it really the agreement is better than I really would have expected. So especially if you take into account that these intake numbers are reportedly based on about 20% of our dietary intake. So you could sort of multiply this by 5, might be a reasonable overall estimate. And if you multiply this by 5, you're almost exactly this on the right. I think that's more luck than accuracy at this point in these relative fields. But it just goes to show you that they're within an order of magnitude anyway, and it seems like it sort of validates the general numbers from both studies by being able to come to this sort of an agreement. We also looked at sort of a worst case scenario in that previous video where we said if we take the 95% confidence interval from the intake numbers, that went up to 9.4 billion particles and 6.2 grams per year. Well, the highest individual overall output, stool content from the current study works out to 26 million particles and 17 milligrams per year. So this 95% confidence interval really seems to be a lot higher than anything that's been observed experimentally. These results are one of, I've seen about 10 or 12 papers in the field. There are variations, but they're all generally in this sort of range in terms of particles found per gram. I felt like this was one of the more elaborated studies and had some of the more trustworthy results, but you could do the same sort of analysis for probably a dozen other papers and come to a similar number. So this number 1 milligram per year is one that I carry in my head with me. It seems to be pretty good. It may be off by a bit, but probably not by more than 1 or 2 orders of magnitude, let's say. And at this time in this field and all the assumptions that go into all these studies, 1 or 2 orders of magnitude is pretty useful. Okay, that's really about all I had for this paper. Again, I really enjoy this more where the data are valid, the procedures are valid, and you can look at things like this and learn from the papers as opposed to just having to discard them because they didn't do things right in the first place. So I thank the authors for this work, and I do hope they follow up with a bigger study, and I hope they do more controls and better controls when they do, and learn from that as well, and give us a really more solid picture about whether we can control our microplastic content in our bodies by our behaviors. Thank you for listening, and there's more to come.